Good morning everyone. Today we are going to continue with the connective tissue topic. Already in our last video we have seen the components of connective tissue. Now let's see the types of connective tissue. Please watch the previous videos on cells and fibers of connective tissue before continuing with this video. First let's see what are the different types of connective tissue. The different types of connective tissue are loose connective tissue, dense connective tissue and mucoid tissue. This dense connective tissue is again classified into two varieties. One is dense regular and other one is dense irregular type of connective tissue. Let's learn how to draw loose areolar tissue. While drawing loose areolar tissue, we have to introduce all the cells and fibers which we have learned so far. First, before shading the ground substance, we have to draw the adipose cells because the adipose cells, the fat globules, will be washed away during histological preparation, hematoxylation preparation. So we have to draw the empty cells with peripherally placed nucleus. So this is the place where we are supposed to put nucleus. Then continue with the shading for ground substance. Now let's start drawing the fibers. First, whatever fibers we have learned so far, all the fibers we have to introduce. The first fiber what we are drawing here is the collagen fiber. Collagen fiber, as we have already mentioned, it runs as wavy bundles. So individual fiber will never branch. You can see here only the bundle branches. You can see the branching here. The bundle in the fiber, the complete bundle will branch. Like this we can complete multiple bundles of collagen. Then we can draw the elastic fibers. Here, as we have already mentioned, draw the elastic fibers where the cut ends will curl and there will be branches. Next we can draw the reticular fibers where we can use our lead pencil. There will be multiple branches. There will be straight small fibers with multiple branches. Next we can introduce the other cells. Only cell what we have introduced so far is adipose cells. Now we can introduce other cells. First what we are drawing is the plasma cells. We can see these are oval cells. We can draw the cytoplasm. Then we can draw the nucleus. You know the nucleus of the plasma cells are cartwheel shaped. So peripherally placed nucleus with cartwheel appearance. The chromatin is arranged in the form of a cartwheel. So we can see in this picture it is arranged in the form of cartwheel shape. Next cell what we are drawing is eosinophils. So where we can draw multiple eosinophilic granules and we can see the nucleus is somewhat kidney shaped nucleus. So we have made the kidney shaped nucleus and there will be multiple eosinophilic granules. This is eosinophils. Next we are drawing the neutrophils. So they are abundant. We can draw multiple. Here I am drawing only two. Actually we can draw many. The cytoplasm is drawn and the nucleus is segmented. There will be multiple segments in the nucleus. The next type of cells what we are going to draw is the basophils. These are rounded cells with many basophilic granules. First we will shade the cytoplasm, then we can mark many basophilic granules. And the nucleus is bilobed or it is somewhat kidney shaped nucleus. So next we can draw after drawing the granules, we can draw the nucleus which is somewhat kidney shaped large nucleus. So this is how we have to draw the nucleus for basophils. Now the next type of cell what we are going to draw is small lymphocytes. These are very small. The cytoplasm is very minimal when compared to the size of the nucleus. So this is how we have to place the nucleus. It occupies most of the cell. Next, this is large lymphocyte where the cells are a little larger and the nucleus is multi-segmented. So there will be multi-segmented nucleus in a larger cell. Next type of cell what you are going to draw is the monocyte. These are larger cells. These are nothing but the largest leukocytes which has got a kidney shaped nucleus. You can see how we have to draw the kidney shaped nucleus. Next we are going to draw macrophages. These are large cells which will have foamy cytoplasm and it will have large irregular nucleus. So what you are going to draw is large irregular nucleus, and it also, the cytoplasm also will have some inclusions, engulfed foreign particles. The last type of cell what we are going to introduce here is mast cells. These are rounded or oval cells which will be having a centrally placed nucleus, and there will be multiple granules, multiple hematoxylin granules or multiple basophilic granules, which will be so numerous it can even mask the appearance of mask the nucleus. So what you are drawing is the granules. This is loose areolar tissue. Next, let's draw the dense connective tissue, the dense regular connective tissue. The same way we have to shade the entire field. So this is nothing but the ground substance. So we have to shade completely the entire circle without leaving a gap. So the ground substance is completed. Dense regular connective tissue is nothing but packed collagen fibers arranged in a regular fashion. So all the features of collagen fibers has to be there. These are wavy bundles. The individual fibers will not branch, only the bundles branches. So the entire bundle can branch, the individual fiber will never branch. So now we can see the collagen fibers which branches, collagen fiber bundles which branches, the single fiber will never branch. So in the same way we have to complete the entire slide without leaving any gaps. So the gaps also have to be completed with the next bundles. These are parallelly arranged bundles. All these collagen fibers are usually secreted by the fibroblast. So at the end we have to draw some fibroblast nucleus and also some fibroblast which has space inside in between the spaces. Because of these parallelly arranged fibers, the fashion of the fibers are parallelly arranged, it has got a great strength, great tensile strength. So this type of dense regular connective tissue is found in mainly tendons and ligaments, where it needs a lot of strength for the movements of the muscles and for holding the joints in place. So after completing the entire fibers, we can draw few fibroblasts. So there will be multiple processes for the fibroblasts. So we can draw few processes and shade the cytoplasm. These fibroblast cells are the one which is going to secrete the fibers which are seen here. Now let's place the nucleus for the fibroblast. These are placed in the center of the cell, and also we can place some nucleus in between these fibers. These nucleus also represent the fibroblast nucleus. So this is the dense regular connective tissue. Next, let's draw the dense irregular connective tissue, where in the same way we have to shade the entire field which represents the ground substance. Next, here also we are going to draw the collagen fibers. Only the difference in the arrangement is the collagen fibers here are arranged in different direction. So we can draw the collagen fibers in different direction. So some are parallel, some are vertical, some are horizontal, and some also runs transversely. So those fibers which are transverse are finally drawn with these kind of dots. So the cut ends only we can see in transversely arranged, in longitudinally arranged fibers. So the longitudinally arranged fibers, we can see only the cut ends. So we have to fill the entire field without leaving much gaps with these multiple different directionally arranged collagen fibers. Since the fibers are arranged in different direction, it can withstand the force which are acting from different directions. So it is mainly seen in the dermis, the reticular dermis. It is also seen in periosteum of bone and it is also seen in the joint capsule, etc. Finally, after the completion of all the fibers, we can place the nucleus of the fibroblast. So these are the fibroblast nucleus. These are predominant cells which are seen in the dense irregular connective tissue which secretes these fibers. The next type of connective tissue is the dense irregular connective tissue. Here also we have to draw first the ground substance. The ground substance is mainly made up of glycosaminoglycans, which is a viscous solution on which we can see some fibers, the collagen and reticular fibers. Next we can see special type of mesenchymal cells which are stellae shaped cells. This is how it will have many cytoplasmic processes. So main example for mucoid connective tissue is the umbilical cord. So inside the umbilical cord, the jelly like substance which is present is the mucoid connective tissue. That is called as Wharton's jelly. The mucoid connective tissue of umbilical cord is called Wharton's jelly. So this is how we have to complete the mucoid connective tissue. This marks the end of connective tissue series. Thank you.